Hi, and welcome to the review on drawings from Chapter 6 and Option D. Again, Chapter 6 and Option D are both studies in human anatomy and physiology. This series covers drawings only. It does not include things that you need to know like processes, enzymes, physiology, and other facts that you're responsible for. Some of these drawings are required. In other words, you may be asked to draw them on an exam. For the required drawings, I'm going to put a red star on them, meaning that you should know how to draw and label them. You also may be asked to annotate some of those features with their functions. Other drawings are just good to know. So in other words, things that you should be able to visualize in order to answer questions about them. The drawings from Chapter 6 and Option D are broken down in this order. Okay, we're going from digestion and absorption, further studies in digestion, further studies in the liver. Then the blood and transport system, the human heart, the immune system, the ventilation system, neurons and synapses, hormones and homeostasis. And then these two topics are part of the higher level content for the option, D5 on hormones and D6 on the transport of respiratory gases. There are no drawings from Chapter D1 nutrition, so that's something that you'll have to review in some other way. Okay, the first drawing we're going to start with is the human digestive system. The human digestive system starts in the mouth. You don't need to draw the whole face, just the mouth is fine. And the mouth leads into a tube called the esophagus. I'm going to go back and label these later. The esophagus widens into the stomach. It's important to note that there's nothing separating the esophagus from the stomach. Okay, it's just one continuous muscular tube that widens and narrows in certain spots. The stomach then leads into the small intestine. And I'm going to stop drawing the small intestine for just a moment so that I can show some ducts that are going to empty into this first part of the small intestine. One of these ducts comes from the pancreas, and the pancreas is kind of tucked over here. It's a bumpy organ that kind of looks like this. It's got a bunch of important jobs, one of which is to secrete hormones into the bloodstream, which we'll get into later, and the other that you need to know is that it empties pancreatic juice into the small intestine. Joining this duct is the gallbladder, and the gallbladder is going to empty into this same duct as the pancreas. So something like this. Okay, and then they're both going to join and empty into this first part of the small intestine. Okay, now the gallbladder is part of the liver. Okay, so I'll give you just a second to catch up there. The small intestine, is a narrow, long, curvy tube, also called the ilium. Okay, and the small intestine then widens into the large intestine. And the large intestine has three parts: the ascending, the transverse, and the descending. Okay, also known as the colon. And of course, they empty out through the anus. Okay, so I'm going to go back and label these now. So we started in the mouth. The mouth widens into the esophagus. The esophagus widens into the stomach. The stomach leads into the small intestine. Okay, emptying into the small intestine are juices from the pancreas and the gallbladder. The gallbladder secretes bile, which of course is produced by the liver. Okay, and then the small intestine leads into the large intestine and any material exiting the large intestine is going to be egested through the anus. Okay, so just to go back and talk about a couple things here, this is one of the drawings that you absolutely must know how to draw and label. You may also be asked to annotate with some of the functions. I'm not going to write them, but I will go over some of them. In the mouth, we're going to see the secretion of salivary amylase. That salivary amylase is going to break down starch into a disaccharide called maltose. It also, that saliva also moistens the food. Once food reaches the stomach, okay, we're going to have a couple things going on. That low pH is going to kill any pathogens that are in the food. And that low pH is also going to activate an enzyme produced in the stomach called pepsin. And pepsin is responsible for the breakdown of proteins. When that food exits the stomach, it's going to enter the small intestine where it meets some juices secreted by the pancreas. Those pancreatic juices are going to buffer that acidic food that's leaving the stomach. It's going to turn it in from a low pH to a more neutral pH because the pancreatic enzymes need a neutral pH in which to operate. Some of those pancreatic enzymes include pancreatic amylase, which is, again, responsible for breaking down starch into the disaccharide called maltose, and lipases, which are going to break down lipids. Speaking of lipids, those lipids are emulsified, okay, by the bladder, which is produced in the liver but is stored in the gallbladder. Did I just say the enzyme bladder? I meant the enzyme bile, which is secreted by the gallbladder. And let's see, into the small intestine that food will go. We're going to see the continuation of the enzymes breaking down those large molecules into smaller molecules. Those small molecules are absorbed into the bloodstream by the small intestine. The large intestine, generally speaking, is just for water reabsorption. And that about does it for the human digestive system. Okay, the next drawing is for a cross-section of the ilium, also known as the small intestine. This is one of the drawings that you absolutely must know how to draw, label. You may also be asked to annotate with some of the functions. Okay, so my small intestine, I'm going to see a couple of different layers. Okay, it's generally a circular tube. Okay, and it's got a couple layers of different types of muscle tissue. The very outer layer is made up of longitudinal muscle. Okay, so the longitudinal muscle is going to run the length of the small intestine. It's responsible for moving food through the small intestine through that muscle action called peristalsis. The next layer, we're going to see a different muscle layer, and this is the circular muscle. Okay, and this muscle squeezes the small intestine. It works in conjunction with the longitudinal muscle to both squeeze the food and move the food down through the small intestine. On the inner layer of the small intestine are a bunch of finger-like projections. Okay, and these finger-like projections are called a villus. Villus being singular, okay, villi being plural. So each one of these is called a villus. They're there to help increase the surface area for absorption. We'll talk more about that in the next picture. And the inner part of the small intestine is called the lumen. This is the part where food actually passes through. Okay, this next drawing is from 6.1. It is the cross-section of an intestinal villus. Again, this is definitely a drawing that you should know how to do, and again, you may be asked to annotate some of these features with their function. All right, so the villus, again, is one of the finger-like projections in the small intestine. Down beneath it would be that circular and longitudinal muscle. On the outside of the villus are a row of cells, one cell thick, okay, so a very thin row of cells. Okay, and these are called epithelial cells. Okay, they're on the outside of this villus. And these epithelial cells are covered with these hair-like projections. These hair-like projections are going to help increase the surface area even further, and these are called microvilli. These are really important to know because if you're asked about some of the structures of the villus that help with digestion, it's really important that you mention that the microvilli help to further increase surface area for absorption. Okay, so now let's talk about where those absorbed nutrients are going to go. Again, you don't need to draw this part, so I'll draw this in blue. Okay, again, food is sitting here in the lumen of the small intestine. It's being broken down by those enzymes into really small molecules. Okay, those small molecules are then going to diffuse or be pumped into these epithelial cells and then into the villus. So what happens to them once they're in the villus? Well, they're going to get picked up by one of two main types of vessels. One of them is called the lacteal. The lacteal is part of your lymphatic system. It's responsible for absorbing and transporting those digested lipids. Also, our villi are highly vascular, meaning that they contain lots of blood vessels. So there's a blood vessel running right through this villus, and it's connected with many capillaries. Okay, so I'm going to label one of these capillaries. Okay, so it's important to note that these capillaries, again, their function is to absorb the nutrients into the bloodstream and then transport them to tissues. Okay, so this isn't necessarily a structure, but this represents the blood flow in, and in this part, this represents the blood flowing out. Whoops, that looks a little messy there. Blood flowing out. Okay, and I believe that just about does it for our villus. Again, make sure that you're understanding and being able to note the functions. The microvilli help increase surface area for absorption. The capillaries are going to absorb those digested nutrients into the bloodstream. Okay, the epithelial cells are one cell thick, making it easier for molecules to move into that bloodstream, or into the lacteal, which again is part of the lymphatic system and is going to transport fats. Also of note here is what's on the inside of these epithelial cells. These epithelial cells are full of mitochondria, and those mitochondria are going to help generate ATP, which we need for the active transport of food molecules into these epithelial cells and then into the bloodstream. And that'll be on my next drawing. This next drawing comes from D2, which is part of the option, okay, on digestion. And it is a required drawing for that section. Over here is a really poor drawing of a villus of the small intestine, and what we're going to do in this drawing is zoom in on a couple of those epithelial cells. Okay, so if I'm drawing these three epithelial cells, I know these are kind of up and down. I'm going to draw them side to side. Okay, and I'm going to go ahead and just kind of draw three of them together like this. Okay, now up here would be the lumen of the small intestine. Lumen, wow, that's really bad. The lumen of the small intestine. Okay, and down here would be inside of the villus. And inside of the villus, again, you're going to find a capillary network and the lacteal, and their job is to absorb the food that is coming from this lumen into either the capillary, into the bloodstream, or into the lacteal, into the lymphatic system. So all of these features of these epithelial cells are to help these cells get the food molecules, get those digested nutrients from the lumen into the either the bloodstream or the lacteal. One of the features that helps them do that are these microvilli. Again, these microvilli are going to increase the surface area for absorption. In order to increase the surface area for the into either the bloodstream or the lacteal on the inside of the villus, we're going to see actually kind of like a curvy section of the membrane. And these curvy sections are called invaginations. Okay, and those invaginations, again, just increase the surface area for diffusion of materials from the epithelial cells into the villus. Obviously, these epithelial cells are eukaryotic, so they're all going to include a nucleus, although that's generally not a required part of these drawings. It's just kind of good to know. An important, very important feature of these epithelial cells, as I mentioned in the last video, are mitochondria. So inside each one of them, I'm going to draw a mitochondria with its double membrane and its matrix. These mitochondria are very important. They generate ATP for active transport in case there are molecules in the lumen that need to be pumped into the cell or then pumped out of the cell into either the capillary network or the lacteal. If you'll notice here, these cells are very close together, and this is what we call a tight junction. The tight junction allows, actually it disallows food particles from diffusing from one epithelial cell into the other. What we want is for food to go from the lumen into the epithelial cell into the inside of the villus. What we don't want is it jumping from epithelial cell to epithelial cell to epithelial cell, and these tight junctions really discourage that. And those are the epithelial cells of the villus. Next up is the gastric pit. This is not a structure that you need to know how to draw, but you may be asked to identify different sections of the gastric pit or features of the gastric pit. So it's one that you really ought to know. On the inside of the stomach, okay, remember the stomach is a muscular wall, and on the inside we're going to find a mucus layer. If you were to really zoom in on that muscular layer, you're going to find millions of holes called gastric pits. So we're going to draw one of them. Okay, again, just to let you know where we're at, this is the muscular wall of the stomach. Okay, in here is the lumen or inside of the stomach. And, okay, surrounding this muscular wall is the mucus layer of the stomach. All right, now lining the gastric pit is a whole series of cells, and these cells may look identical, but they're not. Okay, these different zones of cells do different things. So I'm just going to draw a dot in here to represent the nucleus so that you can take my really poor drawings, and understand that each one of these is an individual cell. Okay, this first zone of cells that's nearest to the top of the gastric pit, these are our mucus secreting cells. That's an O. Mucus secreting cells. And obviously their job is to secrete mucus. They're up near the top of the gastric pit because that's where the mucus layer needs to be. In this middle zone of cells, these are our pepsinogen secreting cells. And you guessed it, these cells secrete pepsinogen. Pepsinogen is a protein that, when activated, turns into the enzyme pepsin, and pepsin is then responsible for breaking down large polypeptides into smaller polypeptides. So what is it that activates this pepsinogen? Well, that pepsinogen is activated by hydrochloric acid, and that hydrochloric acid is secreted by these cells all the way down here at the bottom, okay, called the HCl or hydrochloric acid secreting cells. And again, that hydrochloric acid is going to do a couple of things. It is going to activate pepsinogen. Pepsin only works in an acidic environment. And that hydrochloric acid is also going to break down or destroy any pathogens that might be in the food. And that is the gastric pit. Okay, the liver is the largest organ in your body, and it does a lot of really important things in terms of regulating nutrient levels in the blood. In terms of blood flow, we've got two major blood vessels leading into the liver and one major blood vessel leading away from the liver. Okay, so this first blood vessel is called the hepatic portal vein, and it brings in blood from the small intestine. Now, this is one of the most interesting blood vessels in the body, mostly because the blood that is flowing through this hepatic portal vein varies wildly in terms of nutrient content. During times of digestion, that blood may be very high in certain nutrients, and in other times when we're not eating, may be very low. This blood is deoxygenated and flows into the liver. Okay, so that is the hepatic portal vein. This blood vessel brings in blood from the heart, specifically through the aorta, and this is called the hepatic artery. The hepatic artery. And this again is bringing in oxygenated blood from the heart to the liver. In the liver is a series of tubes that are kind of like capillaries, but they are called sinusoids. Okay, we'll cover that in the next drawing, but these sinusoids are going to kind of process the blood from the hepatic portal vein and the hepatic artery. They're going to do a lot of magical things, and out from the liver is going to come a bunch of deoxygenated blood through this blood vessel called the hepatic, hepa meaning liver, vein, and this blood is going to go to the heart via the vena cava. And this blood has a very steady level of nutrients. Okay, so the job of the liver is to regulate the nutrient level in the blood so that the nutrient level in the blood leaving the hepatic vein is constant. Okay, this is a drawing of one of the many sinusoids of the liver. Again, they are capillary-like tubes that are going to serve in terms of regulation of the blood flow in the liver. So now might be a good time to kind of pause and draw the structures that I already have on here. Okay, and the blood coming into these sinusoids is going to, again, if you remember our last drawing, be from two places. One of them is from the hepatic portal vein, Could be high in nutrients. It's deoxygenated though. Okay, and then we have blood from the hepatic artery carrying the oxygen that, artery, carrying the oxygen that our liver cells need in order to perform all of their functions. All right, inside the sinusoid, a bunch of things are going to happen, and then eventually that blood is going to leave through the hepatic vein. All right, that's all stuff that we already knew. So let's talk about what some of these cells that are pictured in this sinusoid are. The walls of the sinusoid are made up of cells called hepatocytes. Okay, hepatocytes are also known as liver cells. They're going to function in terms of changing toxins into forms that are water soluble so they can be passed out through the urine. They're also going to be responsible for the production of bile and some other really important things that you need to review in chapter D3. These cells lining the inside of the sinusoid are not epithelial. Epithelial is on the outside. These are endothelial cells. Okay, so they're going to help move things in and out. And then these gaps in between the endothelial cells are called fenestrations. Those allow for larger molecules, okay, to move into the walls of the sinusoid, also called the hepatocytes. These irregularly shaped cells are the Kupffer cells. Okay, and these Kupffer cells are going to be responsible for the breakdown of these old erythrocytes and for the breakdown of the hemoglobin that is inside of them. And that is the sinusoid of the liver. Okay, hemoglobin is a very important molecule. There are a bunch of things that you need to know about hemoglobin. It's a protein, so I'll go ahead and list these here, even though it's not technically part of the drawing. It has a quaternary structure, meaning it's made of many polypeptides that are joined together. And I'm going to find this in our erythrocytes, okay, also known as our red blood cells. It's also important to note that you may see questions about sickle cell in regards to hemoglobin. Okay, so like I said, hemoglobin is made up of 4 polypeptides, which I'm going to just denote as this. So there's 1, 2, 3, 4. Okay, 4 polypeptides. In the middle of each polypeptide is what we call a heme group. And this heme group is where the oxygen molecule that the hemoglobin is responsible for carrying is actually going to attach. The heme group. In the middle of each heme group is 1 iron atom. Hold on a second. Here we go. 1 iron, or Fe for you chemistry nerds, atom. And this is where the oxygen is actually going to attach within the heme group. And this whole molecule would be called hemoglobin. In terms of those Kupffer cells in the liver, if you studied the D option on further anatomy and physiology, when those Kupffer cells are breaking down the old erythrocytes and the hemoglobin molecules, different things are going to happen to each one of these parts of hemoglobin. Polypeptides can be recycled into individual amino acids, which we need during protein synthesis. This heme group is going to be turned into bilirubin, which is one of the ingredients needed to make bile. And this iron atom is going to return to the bone marrow, where those new red blood cells are synthesized. So we're going to need iron to rebuild more hemoglobin molecules. And that is hemoglobin. Okay, in order to be able to draw or recognize artery and vein cross sections, it's important to understand that arteries and veins have very different functions. Arteries carry blood away from the heart, A for artery, A for away. Veins carry blood towards the heart. Arteries carry blood at a high pressure, which directly relates to their structure, and veins carry blood at a low pressure. Veins also have valves to prevent backflow, so that blood doesn't flow in the wrong direction. Arteries don't have those. I know most people are tempted to say that arteries carry oxygenated blood and veins carry deoxygenated blood, but that's not always true. Go back and check out those pulmonary arteries and veins for more information on that. So arteries, since they carry blood at a high pressure, are going to have a very, very thick outer wall. Okay? And this thick wall is made of muscles. That says thick. Thick muscular wall with stuff called collagen. Okay? Collagen with an E. And that collagen makes it elastic. Okay, so we have a thick muscular wall that has collagen in it and it's very elastic. That makes the walls of the artery very flexible but also strong and able to withstand that high pressure. It also has a relatively narrow lumen or opening on the inside, and that's so that we can maintain the pressure in the arteries. Remember, those arteries are coming from the heart and they need to be able to carry blood at a high pressure. Those ventricular contractions have to create a lot of pressure so that blood can reach all of the distal tissues. Veins, on the other hand, have a very thin wall. They're not carrying blood at high pressure. Okay, you're not going to be asked to draw this, so this type of kind of squiggly, squaggly drawing is okay. It's more likely that you'll have to recognize them based on a cross section. Okay, but here we have a very thin wall because it's not carrying blood at a high pressure, and this is a wide lumen. Okay, so very different than those arteries. Okay, there are many ways to go about drawing the human heart, many different methods. You're going to be taking your IB exam in pen. It's important to draw in pen because your IB exams are actually going to get scanned in by a computer and pencil lines aren't going to be picked up. So we can't rely on being able to erase, okay, any lines that are drawn because we're going to be drawing with pen. So I'm going to draw the human heart 1/2 at a time. Okay, I'm going to draw the right side first, and then I'm going to draw the left side. I'm going to start at the top right-hand side of the heart. Again, notice that these are reversed because we're looking at someone's heart, so it's their right and left, not ours. Okay, and I'm going to start with the blood vessel that leads into the heart. This is the vena cava. You're not required to draw the superior and inferior vena cava. This would be the superior, it's coming from the top, but we're just going to draw one for now. This blood vessel leads into the right atrium, and this right atrium is going to lead into the right ventricle. This right ventricle is going to have a blood vessel coming out of it. We'll talk more about that in just a minute. Okay, and so here is the rest of our right atrium and right ventricle. Notice that I'm leaving a little bit of a gap here. Okay, that's going to be for one of our valves. Okay, all right, this valve looks like this, and it's important that they're pointed this way because when they are pointed that way, you can clearly see which direction they are opening in, okay, and which direction, if blood were to push against them, they would close. That tells your IB examiner a lot about your knowledge about the blood flow in the heart. I also have another set of valves that look like this. Okay, these are the semilunar valves. Again, it's important to know which direction they open so that you can draw them at the proper angle. They're angled up, meaning blood flow is going to be flowing up in that direction. All right, so time to do some labeling here on this side of the heart. This is the vena cava, and the vena cava is going to bring blood in from all over the body, okay, deoxygenated blood returning to the heart. This is the right atrium. Okay, that receives that blood. The blood is going to go through a valve called the right atrio. I don't have enough space here, it's all one word, but atrioventricular valve. There are all kinds of names for this valve. I like to call it the atrioventricular valve because it tells me what side of the heart I'm on and what two chambers it separates, the atrium and the ventricle. This is the right ventricle. Okay. When blood flows through this right ventricle, when this right ventricle contracts, it's going to push blood through these valves called the right semilunar or half moon valves. And that blood is going to go through this blood vessel called the pulmonary artery. It's leading away from the heart, so it is an artery. Pulmonary meaning towards the lungs. It's deoxygenated blood. It's going to go to the lungs. It's going to pick up some oxygen, okay, and it'll return back to the heart later. One of the things that we should definitely note in here, notice how the atrium is smaller than the ventricle. And I'm also going to kind of shade in this ventricular wall here. The ventricular wall is thicker than the arterial wall because the atrium, okay, doesn't have to make as strong of a contraction. It's only pushing blood from the atrium into the ventricle. The ventricle, on the other hand, has to have a much stronger contraction because it's pushing blood all the way up to the lungs, so it needs a thicker muscular wall. All right, now I'm going to work on the left side of the heart. Okay, the left side of the heart, I'm going to draw this backwards. Okay, so there's a blood vessel leading, again, away from this part of the heart. Okay, so I want to make sure that I draw that so that none of my lines cross. Okay, and here's going to be my left ventricle and my left atrium. Again, I have a blood vessel leading into my left atrium, so I want to make sure that I draw that. I'm going to draw this wall that separates the atrium and the ventricle. I'm going to draw my valves, again, pointing in the direction of blood flow. Blood is going to flow from the atrium into the ventricle, so I want to make sure that that's the direction my valves are pointing. Same thing with the valves down here. There's another set of semilunar valves right here. Okay, again, kind of pointing up, we can see that the blood is going to be flowing up from that ventricle into that blood vessel. So let's go ahead and label some of these things, okay? Again, when the blood is when the blood is flowing through the pulmonary artery, it's going to go to the lungs, it's going to pick up oxygen, and it's going to return to the heart via this blood vessel called the pulmonary vein. Okay, vein because it's heading towards the heart. What makes this vein different is it's carrying oxygenated blood. That pulmonary vein is going to empty into the left ventricle. That blood is going to flow through the left atrioventricular valve into the left ventricle. When the left ventricle contracts, it is going to push open the left semilunar valve. And let me just do a little bit better job of writing that here. Left semilunar valve. Okay, into this giant blood vessel here called the aorta. A-O-R-T-A. Aorta. And that aorta is then going to branch off into several branches, into all kinds of arteries that carry blood throughout your body. Lots of pressure here. The way we create all that pressure in the aorta to make sure that oxygenated blood reaches all our tissues is by a very strong left ventricular contraction. Because that ventricular contraction is so strong, this left ventricle has a very thick wall, much thicker than the left atrium, and even thicker than the right ventricle. And that is the human heart. If we wanted to trace the blood flow, again, I don't think you would be asked to do this in a drawing, but just in case you've had to mentally trace the blood flow to answer a question, deoxygenated blood comes from the rest of the body through the vena cava into the right atrium. From the right atrium, okay, that right atrium is going to contract and push open this right atrioventricular valve. When that blood fills up the right ventricle, okay, the right ventricle is going to contract. That contraction is going to push these right atrioventricular valves closed, and it's going to cause the right semilunar valves to open. Blood is going to go through those right semilunar valves into the pulmonary artery, where it's going to head to the lungs. It's going to pick up a bunch of oxygen at the lungs. It's going to re-enter the heart through the pulmonary vein. Once in the pulmonary vein, it's going to enter the left ventricle. When the left, whoa, I just found a major mistake here. I hope you found this earlier. This is not the left ventricle. This is the left atrium. Sorry about that. So as I was saying, when the blood returns to the heart via the pulmonary vein, it's going to enter the left atrium. When the left atrium contracts, it is going to push open these left atrioventricular valves and enter the left ventricle. When the left ventricle is filled with blood, it is going to contract. That contraction is going to push these left atrioventricular valves closed, and it's going to push these left semilunar valves open. Then that blood is going to flow through the aorta into the rest of the body. And that is the human heart with the blood flow. Okay, this next topic is control of the heartbeat. Now, you're not going to be asked to draw this. It's just one of those things that's nice to visualize. Again, so I'm going to do two types of visuals here. I'm going to do a very bad and very simple drawing of the human heart. Okay, so we have our right atrium, our right ventricle, our left atrium, and the left ventricle. Okay, and I'm going to do another diagram over here on this side where we actually write down the organs and tissues responsible for control of the heartbeat. Your heartbeat is actually controlled by a center in your brain called the medulla oblongata. 2 L's. Okay, the medulla oblongata. Wow, hopefully you're a good listener because that handwriting is really hard to read. The medulla oblongata is going to send messages along nerves to a node in your heart called the SA node. The SA node or sinoatrial node is also called the pacemaker. Either is acceptable from IB. Okay, this SA node is located right here in your heart. Okay, this is the SA node. And it's a specialized group of nervous tissue. Okay, and that nervous tissue kind of runs into the walls of both the right atrium and the left atrium. And when the SA node fires, it is going to do two things. One of the things that it does is it makes the left and right atrium, so atria is plural for atrium, it's going to make them contract. Okay, so both the left and the right atrium are going to contract at the same time because they're both connected to the SA node. The other thing that the SA node is going to do is it is going to pass along that signal to another node called the atrioventricular node or AV node. Okay, the AV node is also located in the right atrium. It's right about here. The AV node. Okay, so there's obviously some kind of nervous fiber that runs to the AV node. When the AV node receives the signal from the SA node, it's going to cause a ventricular contraction. Okay, now if you are studying the option D, D4, we need to know the names of these fibers that run through the ventricular sections. Okay, so, and actually that's a really bad drawing of them because they don't run that way. Sorry about that. These fibers are going to run this way and connect to the ventricles. Okay, and these fibers that run from the AV node are called the Purkinje fibers. Okay, and those Purkinje fibers, okay, just connect the AV node to the ventricles. When the AV node fires, it's going to cause a ventricular contraction. So the ventricles contract. Okay, and again, okay, if you are studying option D, you need to know the names of those fibers. Okay, so one more time, here's how your heartbeat is controlled. The medulla oblongata in the brain sends a signal to the pacemaker or the SA node. The SA node makes the atria fire. I remember that by A for SA, A for arteries. Sorry, not arteries, atria. Okay, that SA node is going to also pass along that impulse to the AV node. The AV node is going to send a signal to the ventricles along the Purkinje fibers and cause a ventricular contraction. So the second letter in each of the nodes tells you what contracts. Okay, also judging by this sequence, we should be able to note that it is the atria that contract first and then the ventricles contract. The left and the right atria contract together. Followed about 1/10 of a second behind is the ventricles, and the left and right ventricles contract together. So there's about a 0.1 second difference in between those contractions, and that is what we refer to as the lub-dub. And this is the control of the heartbeat. All right. An ECG or electrocardiogram measures the electrical activity in the heart over time. You will not need to know how to draw one of these, but you will need to know how to annotate one. So let's just briefly review this concept. On the x-axis here, we have time. And on the y-axis, we have electrical activity. Okay, and by electrical activity, this has to do with your SA and AV nodes. Okay, and when they're firing, they are nervous tissue. And nervous tissue works on the premise of electrical voltage. So that's how we're able to measure that. Okay, when we're reading an ECG readout, the first thing we're going to notice is a tiny wave here called the P wave. Don't worry, we'll go back and label that in just a little bit. And then we're going to experience what we call a Q. Oh, this line should be connected here. Q, R, S complex. Okay, that's what we see as this big spike here. Then we're going to experience something called the T wave. And then the cycle is going to start over again. Again, with the P wave, the Q, R, S complex, and another T wave, so on and so forth. All right, so let's annotate what's happening here. This is called the P wave, okay? And the P wave shows the SA node firing. That's an E, so the SA node is going to fire. And of course, we know that when the SA node fires, that both the right and left atrium contract. So we get atrial contraction. Wow, good luck if you can read that. Atrial contraction. Okay, so this is our P wave. Then we have this area called the Q, R, S complex. Okay, and the QRS complex is basically when the AV node is firing, that's Q, okay, and sending that message along the Purkinje fibers to the ventricles, okay. So the QRS complex, okay, is going to involve a couple things. So this is when our AV node fires, sending that electrical impulse down the Purkinje fibers, and this is when we are getting a ventricular contraction. And that makes sense to me because we know the ventricles are contracting with much more force. So it makes sense to me that that wave is bigger, okay? That QRS wave is bigger than the P wave where the atrial contractions take place. Okay, and then we have this area called the T wave, okay? And the T wave, okay? This represents when the nodes are repolarizing. If you've already studied section 6.5, then you know that that means we're kind of recharging. We're resetting those ions on the correct side of the membrane, reestablishing that electrical potential and getting ready for the next heart cycle. Okay, so what we see here from P to T represents one cardiac cycle. So you don't need to draw this, but I'll just circle that for you. This is what we call a cardiac cycle. Okay, and then of course that process is going to repeat itself in the next cycle. P wave where the SA node fires, QRS complex where we see the AV node firing, a ventricular contraction, all that good stuff, and then the T wave where we see repolarization. Okay, chapter 6.3 is all about defense against diseases, and one of the things that I find with students in this section is the confusion of several similarly sounding words, antigen and antibody. Okay, so let's draw what an antigen-antibody complex looks like to help you figure out the meanings of these words. All right, so what I'm going to draw here is a pathogen. Pathogens are any organism or virus that can cause a disease. And like any cell, on the outside of a pathogen are proteins called antigens. Antigens are a membrane protein. Remember, membrane proteins have several different functions, one of which is cell recognition. All cells have antigens. They have proteins on the outside, and that helps your immune system recognize them as self or non-self. So if the antigens match your cells, your immune system is going to recognize that as one of the cells that's supposed to be in your body. If your immune cells encounter this pathogen and they don't recognize these antigens, it's going to trigger an immune response. Okay, so antigen, we need to remember, are proteins that are found on the outside of cells. In this case, this antigen is finding itself on the outside of a pathogen. In the case of specific immunity, one of the last things that's going to happen is the production of something called an antibody. Antibodies are Y-shaped proteins. Okay, this is an antibody. They are produced by your B cells, also called plasma cells, also called lymphocytes. Okay, and the antibody has two binding sites. And this is where the antibody is going to bind to the antigen. Now, this antibody binding site has to match the shape of the antigen. So watch what this is going to look like. Here's my Y-shaped antibody, and this antibody at the end has a binding site that matches, okay, the binding site for that particular antigen. So each antibody can bind to two antigens or to two pathogens with the same antigen. And that's the antigen-antibody complex. Okay, this graph is not necessarily something that you need to draw, but you might be asked questions about it or asked to recognize it. So it's a good idea to kind of understand what's going on in your bloodstream during an immune response. So we're going to draw this graph together. On the x-axis is going to be time. And on the y-axis is going to be antibody concentration in your bloodstream. Okay. Now, when you first encounter a pathogen, you should have no antibodies in your blood. That's because we haven't seen this pathogen before. We don't have an immune response ready to go for this pathogen. So it's important that we're starting out at zero antibody concentration. When we first encounter a pathogen, it's going to take our body, let's start out at 0 here. It's going to take our body a little bit of time to recognize that pathogen and create an immune response. If we successfully fight off the pathogen, okay, that pathogen goes away and we don't need so many antibodies floating around in our bloodstream. So that antibody concentration is going to go down. It does not, however, go all the way down to 0, okay? And that's because we are then ready for our second exposure to that pathogen, okay? The second time we see that pathogen, okay, our antibody concentration is going to go way up. All right, and before I can explain why, let's go ahead and label some parts on this graph. Okay, so here is our first encounter with a particular pathogen. Let's say this is this year's flu virus. Okay, this first hump here in our graph is what we call the primary immune response. Okay, this is your body building up those antibodies. This is going to take a little while. This process is a little bit slow because first we need to be able to recognize that antigen. We need to be able to find the correct B cells that can produce that antibody. Those B cells have to clone themselves. It's kind of a time-consuming process. This part in our graph right here represents any kind of secondary exposure. And this means any exposure after the first one. Doesn't matter whether it's the second, third, 23rd, doesn't matter. Any subsequent exposure to that pathogen. Okay, and then this big spike in antibody production is what we call the secondary immune response. Okay, notice how much steeper that portion of the graph is. Because we already have those memory cells from the last time floating around in our blood, okay, those memory cells, those special B cells, are already in our bloodstream, ready to produce those antibodies needed for that pathogen. Kind of a good graph to know. All right, let's draw out the human ventilation system. Be careful not to call this the respiration system, as you may hear in some other courses. Remember, respiration is a biochemical process that starts in the cytoplasm and ends in the mitochondria of a cell. Okay, the ventilation system is more of a physical process. Okay, now that's going to begin in the mouth. So I'm drawing someone's head as they're facing sideways. We'll go back and label this later. Or it could be in the nose. We can breathe through a mouth or a nose. And like the digestive system, the mouth leads into a long tube. But this is not the esophagus. This is a different tube called the trachea. So you actually have two tubes leading from your mouth down into your neck. One is the esophagus, the other one is the trachea. They're kind of separated by a little valve called your epiglottis, but you don't need to know that. Okay, so you have your mouth and your trachea. This trachea branches into two large tubes called bronchi. Bronchus singular, bronchi plural. These bronchi are going to branch into smaller branches, much like a tree has a trunk that has then large branches, small branches, and twigs, okay, branching off of it. Those are called bronchioles, and bronchioles end in these air sacs called alveoli. You have millions of alveoli in your lungs. Okay, we're only drawing 6 small groups of them because that would be unreasonable to ask me to do that with this iPad. Okay, now these make up your 2 lungs. Okay, so I usually draw them in there, something like that. And then you have a large muscle underneath of your lungs called the diaphragm. This is one of the muscles that controls your breathing. Okay, so there are other muscles that control your breathing too, but to draw them in here would complicate things. You can stop here for your IB drawing. Okay, but just to give you a point of reference to talk kind of like about where we're at, if this is the nose, then here would be the face and the lower jaw. So there would be an eyeball and like the nose. I know this is really bad. Okay, and then shoulders, arms and hands. Wow, this person has a really small head. Okay, and then there's their abdominal cavity. All right. Again, though, you don't need to draw that. So for my purposes, I'm going to kind of get rid of that so it doesn't mess up with our labeling. And now's a good time to kind of review IB labeling. You should have clearly drawn lines, not sketch marks. Okay, your labels should be done by straight lines that don't overlap, etc. All right, so here we have the mouth. Okay, and the mouth leads into the trachea. Since you've already covered the immune system, the trachea is lined with mucus secreting cells. Okay, it's one of our mucous membranes. Also has cilia to help keep pathogens out. The trachea branches into 2 main tubes, Singular, they are called a bronchus. Plural, they'd be called bronchi. Bronchi branch into bronchioles. Okay, or bronchiole singular. And these bronchioles end in alveoli, or alveolus singular. Then we have our lungs, and this big muscle down here at the bottom called the diaphragm. You'll need to go back and study about the method of human ventilation, but remember, a diaphragm contraction is going to cause air to be inhaled, okay? And we have other muscle contractions that control expiration. And that is the human ventilation system. Before we draw a magnified alveolus, okay, I want to go back and just give you a little picture as to where we're at here. Bronchioles end in air sacs called alveoli, and each one of them are kind of like circular looking, they kind of look like a cluster of grapes, and they are covered with lots and lots and lots of capillaries, because this is where we're seeing the exchange of oxygen and carbon dioxide. So what we're going to do is draw a magnified alveolus. I'm going to zoom in on one of them and just focus on one capillary, and that's what you actually need to draw. Okay, so heading in from the bronchiole, okay, is our alveolus, and alveoli have a very thin wall. So thin, in fact, it's made up of only one cell. The alveoli wall is one cell thick. These are all individual cells. So I'm going to draw those in. Okay, those are called type 1 pneumocytes. Okay, and the type 1 pneumocytes actually form the wall of the alveolus. Okay, now, next thing I'm going to draw in is the other type of cell that we see in an alveoli, and that's kind of on the inside of the alveoli here. Okay, it also has a nucleus, and that is called a type 2 pneumocyte. Okay. Cyte meaning cell, pneumo referring to the lungs, kind of like pneumonia infects the lungs. Anyways, the type 1 pneumocytes form the wall of the alveoli. They're thin, they allow for easy diffusion. Type 2 pneumocytes secrete a surfactant on the inside of the alveolus to keep the inside of the alveolus from sticking together. We want that thing to be inflated, not stuck to itself. All right. Now, generally speaking, a capillary is going to follow this curvature of the alveoli. And we also have to remember that capillaries are also only one cell thick. So there's one wall of the capillary. Okay, remember the capillary is a tube. So this looks like I'm drawing a cross section of the tube. Okay, and this is a capillary. Okay, inside of the capillary, I'm going to find a bunch of our erythrocytes, these red blood cells. Okay, red blood cells have hemoglobin. Okay, and hemoglobin's job is to carry oxygen throughout the bloodstream. So if these hemoglobin molecules are traveling this way, these are coming to the alveolus, these are then leaving the alveolus. Okay, let's think about where they're coming from. The ones going to the alveolus are going to be low in oxygen. Okay, they're being pumped from the right ventricle, meaning they're coming from the vena cava, meaning they're deoxygenated. They need to get rid of carbon dioxide and pick up oxygen. That's why they're coming to the alveolus, and then they're going to leave, okay, and then flow back in through the pulmonary vein into the heart and then be pumped in through the rest of the body. If we're thinking about oxygen, okay, I'm going to do oxygen over here. We're going to have a very high concentration of oxygen in the lungs because we're breathing in heavily oxygenated air. That oxygen has a very low concentration over here, okay? So high concentration in the air, low concentration in the bloodstream. So some of this oxygen via diffusion is going to diffuse into these red blood cells, hang on to that hemoglobin molecule, and be able to travel out through the rest of the bloodstream. To do that, it's got to pass several membranes. It's got to pass 2 membranes to get into and out of the pneumocytes, 3, 4 to get into and out of the first capillary wall, and then 5 to get into the red blood cell. That's a lot of diffusion. But because of the difference in concentration between here, okay, in the alveolus, and here in the bloodstream, it can do that without using any energy. Same thing is going to happen in opposite with carbon dioxide. And that is a magnified alveolus. Don't feel like you need to draw this diffusion part of oxygen. That was just my way of kind of helping you out. All right, here we're going to draw a myelinated neuron, and as opposed to an unmyelinated neuron. I'll get into that in just a second. Okay, I'm going to start with the cell body, and the cell body kind of, I don't know, looks something like this. Okay, it's got these dendrites on the end, it's got a nucleus, a bunch of other stuff in it. Then we've got this long axon. It could be very long, I guess it could be short. Okay, followed by these axon terminals. We'll go back and label these in just a minute. Okay, since this is a myelinated neuron, it's going to be covered in different cells, and I shouldn't draw sketch lines like this. This is an IB drawing after all. Okay, here's my axon. Okay, it's going to be covered in cells like this. These cells are called Schwann cells. Together they make up the myelin sheath, and the areas where there are gaps in between them, okay, are the nodes that the action potential is going to jump around on during a myelinated neuron action, okay, or saltatory conduction. So again, to go back here, this is called a cell body or soma. Then we have the nucleus. These spots here that receive the impulse are called dendrites. Okay, they receive the impulse. This long part here is called the axon, and it ends in the axon terminals. Okay. These cells that cover the axon are called Schwann cells. And together they make up, those are parentheses, wow. Together they make up the myelin sheath, which insulates the axon so it doesn't lose that electrical potential. And these gaps here are called nodes of Ranvier. And those are where the action potential jumps around on during saltatory conduction. If I were to zoom in on these axon terminals, you don't need to do this, but just kind of to let you know what's happening here. So I'll do this in blue since you don't need to know this. Okay, if I were to zoom in here, the axon terminal is going to look something like this. Okay, there's going to be a short gap before it meets the dendrite of the next neuron. Okay, and then this axon terminal is part of what we call the presynaptic neuron. And then, of course, we have the postsynaptic neuron and the synaptic cleft or gap. Okay, and so once the action potential, which you should always draw into your drawings, action potential is going to go this way. Once it reaches the end, okay, then it would cause those chemical neurotransmitters to diffuse across the synapse, bind to our postsynaptic neuron, okay, and then begin a new action potential in this nerve cell. If we're talking about saltatory conduction, again, you don't need to draw this part. This action potential wouldn't look so straight. It would actually be jumping, okay, from node to node to node before passing in that way. But you don't need to draw that. You just need to draw what's in black and what's in red here with our action potential arrow. Okay, understanding action potentials and what's happening inside of the neural cell during an impulse can be really difficult, but we're going to summarize it using a graph. On the x-axis here is the voltage across the membrane, so how the inside of the membrane compares with the outside of the membrane. During resting potential, we're at about negative 70 millivolts. Okay, it's very negative inside. During an action potential, we can see an increase to up to 30 millivolts on the positive side. Okay, so from going negative to positive. All right, now it's important to remember that during resting potential, and I'm just going to draw this down here. You don't have to draw this part if you don't want. During resting potential is very negative on the inside and positive on the outside. Okay? We've got potassium on the inside and sodium on the outside. And that's going to cause our cell to be negative. This is time here on our x-axis. When an action potential is reached, okay, what's going to happen is these sodium ions, I'm going to redraw this like this. The sodium ions are going to enter the cell through the sodium ion channels, okay, and our potassium ions are still in there. So our cell here is going to become very positive on the inside, okay, so that's going to cause the voltage to go up. Okay, once all the sodium has diffused in, that's going to cause our potassium ion channels to open. So our sodium is still on the inside, but now these potassium ions are going to diffuse outside. That's going to bring our voltage in our cell back down. So negative again. So we're going to come back down here. Okay, it actually goes a little bit past where it should. Okay, but you don't really need to know much about that for right now. Okay. Then eventually, our sodium-potassium pump is going to pump the sodium to the outside and the potassium on the inside, returning things how they should. Again, this is still negative, okay? And so now we're kind of back at our resting potential and ready for the next nerve impulse. So let's go through and label what's happening here. Okay, so at this point, this is called, I don't have a lot of room here, this is our resting potential. It's a T, resting potential. Okay. At this point, oh, I'm getting really running out of room here. Our sodium ion, so Na plus ion channels are going to open. Okay, and that's going to cause lots of sodium to come in, to flood in through our cells. And this is an area of time called depolarization. So I'll do it this way. Depolarization. In other words, we are switching the polarity. We're going from positive to negative. Okay, two things are going to happen here at the top. First thing is that the sodium ion channels are going to close, and the potassium ion channels are going to open. Okay? This is an area of time where we call this repolarization. It's an R. Repolarization, sorry, that's kind of hard to read, where we're returning to our normal state of being positive on the outside and negative here on the inside. Okay? The problem is that we have things on the right or on the opposite side. Sodium is on the inside and potassium is on the outside. We don't really want that. Okay, so then at this point here, we are returning, that's a G, returning to resting potential. Okay, so that's here, and that's going to be through the action of the sodium-potassium pump. Okay, don't forget, I guess I should note down here, once we're ready for that pump to start working, we have to close those potassium ion channels. Because the sodium-potassium pump can't create a potential if those ion channels are closed. And that is our voltage during our resting and active potential. Next up is the hormonal control of glucose levels in the blood. Always, 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 the goal of hormonal control of anything really is to achieve homeostasis. So we're going to keep that at the center of our discussion. Okay? Now, when we say homeostasis, we mean like maintaining a normal blood glucose range. Okay, so let's talk about when your blood glucose levels go up. Okay, so increased blood glucose level is going to occur after meal times, especially if you're eating foods that are high in simple carbohydrates. Okay, this increased blood glucose level is going to cause the beta cells of the pancreas to secrete insulin. Okay, and that insulin is going to cause two things to happen. Okay, the first thing that's going to happen is some of that glucose is absorbed by body cells. Glucose triggers some of the membrane proteins to open, allowing glucose to move from the bloodstream into your body cells. Insulin also causes the liver to convert glucose into glycogen. And that is stored in the liver. For those of you who studied the option in D3, you learned about the hepatocytes and their function in this. Okay, both of these actions are going to result in lowered blood glucose levels. Okay, and that gets us back to this homeostatic level. Again, just to kind of review, you don't need this in your drawing necessarily, but glycogen is many glucose molecules hooked together, okay? So it effectively takes them out of the bloodstream, therefore lowering your blood glucose level. Okay, now let's say on the other hand, okay, our blood glucose level is low. Let's do this in black. Okay, so Here we started with high blood glucose levels. Now let's talk about the opposite, like after long periods of fasting, those kinds of things. Okay, this is going to cause the alpha cells of your pancreas they secrete a hormone called glucagon. The way that I remember this is when all your glucose be gone, you secrete glucagon. Anyways, okay, that glucagon is going to tell your liver to turn some of that glycogen that it stored into glucose. Okay, so it causes liver Okay, so there we are catabolizing, breaking down that big glycogen molecule into individual glucose molecules. Okay, and so what this is going to do is this is going to increase your blood glucose level. Okay, and then again, we arrive back in our homeostatic range. Okay, so here we've got two different hormones being secreted. Glucagon when you have low blood glucose levels, and that triggers glycogen turning into glucose. So breaking down that large glycogen molecule, which is going to increase your blood glucose level. On the other hand, when we have high blood glucose levels, the beta cells in the pancreas are going to secrete insulin. That causes glucose absorption, and it also causes glucose to be turned into glycogen in the liver, therefore being removed from the bloodstream and lowering our blood glucose level. Next up is a drawing of the male reproductive system. In general, I think it's easier to draw the male reproductive system from the side, although you're welcome to draw it from the side or from the front. Okay, I generally start with the testes. Okay, and we're just going to draw one because we're drawing it from the side view. Okay, the testes are where the sperm are actually produced. Sitting on top of the testes is a piece of tissue called the epididymis, where the sperm mature. And leading from this tissue is a tube called the vas deferens. And the vas deferens is the tube that you cut during a vasectomy. All right, I'm going to draw it in this shape for a particular reason. Okay, and I'm going to stop for just a second and draw in a couple things that are underneath. Okay, so right underneath of where this vas deferens kind of curves around is the urinary bladder. It's not technically part of the reproductive system, but it does share some of the same ductwork. Okay, so the vas deferens leads into a gland called the seminal vesicle, and the seminal vesicle is where some of the seminal fluid is added. Then the tube continues into the prostate, okay, where many of the other components of the ejaculate are added, including the high carbohydrate part of the seminal fluid. All right, then that tube is going to exit there. It is also going to join with a tube from the urinary bladder. Now, fear not, the urinary bladder and the ejaculate don't come out at the same time. There are mechanisms to ensure that that does not happen, but they do share one single exit tube called the urethra. Okay, and that crosses over like that. So they do share some ductwork there. Okay, now the urethra exits at the penis. Okay, surrounding the penis, or surrounding the urethra rather, is the erectile tissue of the penis, and that will swell with blood during an erection. This is the internal anatomy, okay? So let's go back through and add the external anatomy. Okay, here would be the skin of the penis. Okay, notice how there's still an open hole there at the urethra. And then this is part of the abdomen. And then we have the skin covering the testes called the scrotum. And then this would be leading to the posterior of the body. Now let's go back and label these things. Okay, so we have this tube exiting here called the urethra. We have the erectile tissue. That's an S of the penis. Okay, then we have the penis itself. Okay, the skin covering the testicles is called the scrotum. Followed by the testis. That's singular. Testes would be plural. This is, again, where the sperm is produced. Next, we have the epididymis. That is where the sperm matures. Okay, and it leaves through this tube called the vas deferens. Okay, and that vas deferens comes all the way up and around the urinary bladder. Okay, and then the vas deferens joins the seminal vesicle. Okay. That seminal vesicle, okay, then also empties into or goes through the prostate. Okay, and then we're showing some of that shared ductwork or shared tubing, okay, with the tube that's leaving the prostate and with the urinary bladder. And that is the male reproductive system. Okay, on to the female reproductive system. So the female reproductive system is a little bit easier, I think, when drawn from the front view, okay, rather than a side view. So first I'm going to start with kind of a triangle shape uterus, okay, and that triangle shape uterus empties into the vagina. Okay, now the uterus is a muscle, so we're going to show the muscle wall. And notice how I added that little hump there, kind of at the point where it narrows. That's kind of important. Okay, so we'll talk about that in just a moment. Okay, this narrowed part is called the cervix, and then again we have the vaginal canal. Lining the inside of the uterus is a highly vascular, which means lots of blood vessels, tissue called the endometrium. And this is what a woman is going to shed during her menstrual cycle each month. Okay, so we've almost got all of the parts of the female reproductive system. It's a little bit more simpler than the male. Okay, all we're missing are our ovaries. And a woman has 2 ovaries. And these ovaries connect to the uterus via a tube called the fallopian tube or oviduct. Okay, so let's go through and label this female reproductive system. So again, we have the ovary. Okay, we have the oviduct, or you can call that the fallopian tube. Either is acceptable. Okay, then we have this thick muscular wall of the uterus. Lining the uterus is that highly vascular tissue that the embryo is going to implant on later on if the woman becomes pregnant, and that is called the endometrium. Okay. The point where this narrows is called the cervix. Okay. And the cervix is just kind of like the lower part of the uterus. Okay. It's the pathway for childbirth. This is what dilates during the childbirth. If you hear of like 8 centimeters dilated, 10 centimeters dilated, they're measuring the width of that cervix. And then we have the vagina. Okay, this is also called the birth canal. Okay, pathway to the outside. Some people you may see in their drawings of the female reproductive system, you may see them shade in the endometrium. That's perfectly fine if you want to do that just to make this a little bit easier to see, but you certainly don't have to. And that's a female reproductive system. Now it's on to everybody's favorite topic, the hormones of the menstrual cycle. In this drawing, we're just going to focus on what these hormones do, and in the next one, we'll kind of trace their levels throughout the 28-day menstrual cycle. So it's important to understand where these hormones are coming from. And these hormones kind of start out in an area of the brain called the hypothalamus. Okay, and the hypothalamus is deep within the brain, and its job is to act on the pituitary gland. All right, so if I'm kind of looking at the brain here, okay, and this is, I don't know, here's a person's eyes, nose, and mouth, okay? In their brain, we've got the hypothalamus kind of like right in here, and it's connected to 2 lobes. Okay, and those 2 lobes are the anterior, or front, and posterior, or rear, pituitary gland. All right, in terms of the menstrual cycle, we're going to be focusing just on the posterior pituitary. Those anterior pituitary glands, they don't really do a whole lot, okay, in terms of the menstrual cycle. So, the hypothalamus secretes a hormone called gonadotropin. We're not going to write all this out. GN for gonadotropin, releasing hormone. Okay, and what that gonadotropin releasing hormone is going to do, it's going to act on the anterior pituitary. Okay. That anterior pituitary has cells that are sensitive to this gonadotropin releasing hormone, and when it senses this hormone, it's going to produce some hormones of its own. So this is kind of like a cascade of hormone production. The 2 main hormones it's going to produce are called LH, or luteinizing hormone, and FSH, which stands for follicle stimulating hormone. Okay, follicle referring to the egg structure. Both of these are then going to act on the ovaries. Okay, so the ovaries are the prime targets of both of these hormones. Now, the ovaries in turn are going to secrete 2 hormones of its own. Okay, and those 2 hormones are estrogen and progesterone. Both of these, estrogen and progesterone, act on what we call the endometrium, that highly vascular lining of the uterus. Okay, and they're going to act on this endometrium just at different times. Okay, so early on in a woman's menstrual cycle, okay, these ovaries are going to stimulate just the production of estrogen. Okay, so this is kind of our early hormone. When estrogen is present singularly, that estrogen is going to stimulate, okay, the hypothalamus and tell it to produce even more gonadotropin releasing hormone, which in turn produces more LH and FSH, which tells the ovaries to produce even more estrogen. Okay, and that's what we call a positive feedback loop. So when estrogen is present on its own, it has a positive feedback loop causing even more production of these hormones. And that causes an increase in the lining of the endometrium. After ovulation, so it's worth noting that progesterone is after ovulation. So after the egg is released from the ovary, okay, we're going to see both estrogen and progesterone present. That's going to further increase the lining of the endometrium, so adding vascular tissue to that endometrium, thickening that place where that embryo, if fertilized, is going to implant. And then we get a little bit of a different scenario. When both estrogen and progesterone are present, then we're going to see a negative feedback loop, okay? Meaning that the presence of both of these is going to tell the hypothalamus, hey, we don't need any more of this. We already have a developed ovary. We don't need you to produce any more of these luteinizing or follicle stimulating hormones. We've already released a mature egg, so we don't need those anymore. Okay, and that's going to, again, decrease those. So that's what we call a negative feedback loop. Since we've already ovulated, okay, in the ovaries, we're going to start to see a structure called the corpus luteum form, and it's basically the empty follicle where that egg was released from. And that corpus luteum is going to cause a decrease in these estrogen and progesterone levels. When these estrogen and progesterone levels drop, we can no longer maintain the endometrium, so the woman will start menstruating, and then that drop in both of these levels, since when both of them are present, it's inhibiting, okay, this process. When these both drop, that's going to trigger the hypothalamus to start secreting gonadotropin releasing hormone again, and then we're going to start to see the stimulation of these hormones again, okay, and developing a new follicle, and we're starting the menstrual cycle all over again. If this is a little confusing to you, don't worry, we're going to cover that again on the next slide. Okay, so let's take a look at these hormones of the menstrual cycle and see how they interact with each other to result in ovulation and menstruation. You're not going to ever be asked to draw this. You may be asked to annotate it, and you may be asked questions about it. So it's a good idea to have a visual in your brain as far as what's going on here. Okay, so we're going to track what's happening here, okay, over the course of time. So this is time. And since a woman's cycle is generally split up into 20 days, okay, here's 0 days. Halfway through would be 14 days. Okay, and the end would be 28 days. It's important to know that the 28th day is also the 0 day, okay, because it's a cycle. Now, we're going to be looking at this in terms of a few different processes. So I'm actually going to split this graph up into fifths. So we can track what's going on with 5 different processes. We're going to be tracking what's going on with the endometrium, especially in regards to its thickness. So how thick, how vascular is that endometrium? the greater the thickness, the more ready that endometrium is to receive a fertilized egg or an embryo. Okay, we're also going to be tracking a hormone called progesterone. Progesterone is secreted by the corpus luteum, which you should know is kind of like the empty space in the ovary where the egg is released from. We're going to be tracking estrogen. Estrogen is released from the ovaries also, but it is as a result of some other hormones which we covered in the previous slide, luteinizing and follicle stimulating hormone. We're going to be tracking what's going on in the ovary, okay, and specifically what is happening with the follicle and with the corpus luteum. And we're going to be tracking the gonad. Well, I should probably spell this correctly. Gonadotropin levels. Okay, and by gonadotropins, I mean LH, luteinizing hormone, and FSH, the follicle stimulating hormone. Those are produced by the anterior pituitary. So maybe I should note that, anterior pituitary. I should probably spell that correctly. Okay, so this is a really complex process going on with steroid production and hormone production in multiple parts of a woman's body. Okay, so I'm going to actually start out with these gonadotropin levels. In the early part of the cycle, okay, we are going to see a steady increase in FSH, or follicle stimulating hormone, and LH, or the luteinizing hormone. And right at about day 14, both of these are going to be at its peak. Okay, if we remember what LH and FSH do, okay, one of the things that they're doing, specifically this FSH, which is this bottom line, follicle stimulating hormone, it's going to cause the development of the follicle. Okay, so I'll note that here as follicle development. Okay, so this follicle stimulating hormone is going to develop that follicle, okay, and that's going to get us ready for this big event at day 14, okay, which is ovulation. Okay, so when that follicle is fully developed, when the follicle stimulating hormone is at its peak, we get full development and ovulation. Now, these simultaneously are triggering an increase in estrogen. Okay, so estrogen is going to follow this, okay, at its peak, okay, because FSH and LH act on the ovaries to increase estrogen levels. If we remember from the previous slide, as estrogen increases, it acts on the endometrium. So we're also going to see a steady buildup, okay, of the endometrium levels. When ovulation occurs, the follicle is then going to kind of degenerate into, let's get my pen back going here, into something called a corpus luteum, okay? And as this corpus luteum develops, it is going to, I guess I should write this here, corpus luteum. Okay, as that corpus luteum develops, it is going to trigger the release of a hormone called progesterone. So progesterone is going to peak when this corpus luteum is peaking in its development, because the progesterone is actually secreted by this corpus luteum. And this progesterone, okay, also stimulates a thickening of the endometrium. Okay, so both the estrogen and the progesterone cause the endometrium to develop. Now, assuming that there is no pregnancy, this corpus luteum starts to break down. Okay, it starts to shrivel and go away. Then it's no longer able to produce progesterone. So that progesterone level is going to drop. Okay, and since the presence of both estrogen and progesterone cause a negative feedback loop to the hypothalamus and anterior pituitary, these LH and FSH hormones are going to drop also. Okay, so again, that's a result of the estrogen and progesterone having a negative feedback loop here. Since I'm not producing LH and FSH, that's going to cause a drop in my estrogen levels. And since progesterone and estrogen are responsible for the lining of the endometrium, when those start to drop, we're no longer able to maintain the endometrium and it's shed as menstrual blood. Okay, so that woman is going to be menstruating because she no longer has this estrogen and progesterone in there. Okay, now all of this starts over again because remember, when estrogen and progesterone are low, that's going to stimulate the anterior pituitary and the hypothalamus to start secreting LH and FSH again. These, when present together, inhibit these. So now that they've dropped, we're going to start to see them in the next cycle build up again. And then all of this starts over. It's important to know that LH and FSH both stimulate estrogen, so all three of those are going to peak at the same time and cause ovulation. When ovulation occurs, then we start to see the development of this corpus luteum. Since the corpus luteum secretes progesterone, that's when that one is going to peak. Okay, and then they all start to decline at the same time, resulting in the decline of the endometrial thickness. Okay, D5 is all about hormones and metabolism. And one of the first things that we need to understand about hormones and metabolism are where these hormones are being secreted from. And hormones are secreted from endocrine glands. Now, you won't be asked to draw this, but you may be asked to label a diagram with certain endocrine glands. So I'm going to draw a really bad person. Okay, like really bad. So we have the head, we have the neck, okay, arms, an abdominal cavity, legs, etc. Okay. Perfect. So somewhere in the brain, okay, deep within the brain, we have two really important hormone secreting glands, and this is the anterior and posterior pituitary. Okay, in the neck, we have this kind of butterfly-shaped organ, okay, with another organ in the middle, okay, and this is our parathyroid. And our thyroid. Okay, don't forget that the pancreas is also a hormone secreting gland. We've got glucagon and insulin. Okay, insulin coming from the beta cells and glucagon coming from the alpha cells. Now, sitting on top of your kidneys, so I'm going to draw two convex, really bad kidneys. Sitting right on top of those two kidneys are your adrenal glands. And you guessed it, they secrete adrenaline, also known as epinephrine. Okay, and then two other ones are going to be located in the abdominal cavity, and it depends really what gender you are. So I'm just going to draw a question mark because this gingerbread looking man that I have here doesn't really have a defined gender. So somewhere in here is either going to be ovaries, if that is a woman, okay, so we're talking FSH, LH, and such, or if it's a male, we're talking the testes. Okay, so mostly testosterone. And these are the major endocrine gland locations in the body. Okay, now let's move on to the rather complicated relationship between the hypothalamus and the anterior and posterior pituitary. Okay, the way that we're going to be looking at this, don't feel like you have to draw this, but if I'm looking at a person, here's their mouth, their nose, and their eye. Okay. And then here is their brain. Okay, now within this person's brain, we have several parts, some of which we're going to talk about, some of which we aren't. Okay, but the hypothalamus is generally right about here. Okay, and we've got two lobes of the pituitary gland here and here. All right, so I'll draw that in a better picture. Okay, so that we can kind of zoom in and see what's going on here. All right, so let's draw the hypothalamus, kind of like this. Okay, this is the hypothalamus. Okay, and then this hypothalamus kind of leads into the posterior pituitary. Posterior meaning behind. So as you can see in our drawing, I'm now drawing this part, which is kind of behind. Okay, and not the part in front. Okay, so posterior pituitary gland is here. The anterior pituitary is kind of a second lobe that looks like this. Okay, so anterior pituitary. Now, the way that the hypothalamus interacts with the anterior and posterior pituitary are a little bit different. So let's do the easiest one first. In the hypothalamus are these things called neurosecretory cells. And they are exactly what they sound like. Okay. These are long neurons. Okay, so I'll go ahead and label those. Neurosecretory cells. Okay. Now, these neuron cells, okay, actually produce the hormones up here, okay, in that cell body part of those neurons. They travel down to the terminal end of those neurosecretory cells, and those hormones are actually released as a neurotransmitter would normally be in a synaptic transmission. Okay, so these hormones are actually built in this cell body up here in the hypothalamus, and then they travel down this way, okay, and are secreted into the posterior pituitary. So any hormone actually secreted by the posterior pituitary was originally, okay, built or assembled in the hypothalamus. Okay, so once those hormones are secreted by the terminal end of the neurosecretory cells, they make their way into the capillary network of the posterior pituitary, okay, and they leave through a blood vessel. Okay, then those hormones are free to act on whatever their particular target cells are. There are a bunch of them. There are two that you should probably know. Okay, one of them is oxytocin. Okay, and remember oxytocin is used in uterine muscles. Okay, and in mammary glands. Okay, the other one that you should probably know, okay, is called ADH, antidiuretic hormone, and you know this one. Okay, this is what makes the kidney tubules more or less permeable to water. So it kind of decides how much water needs to be reabsorbed back into the bloodstream, how much water that's in the filtrate needs to come back into the bloodstream and be conserved rather than being passed on, okay, as urine. Okay, now let's move on to the anterior pituitary. The anterior pituitary is much, much different. Okay, so in the hypothalamus, there are still some neurosecretory cells. But instead of extending down into that anterior pituitary, they secrete hormones called releasing hormones into a blood vessel. Okay, and these blood vessels are called portal blood vessels. Okay, this portal blood vessel, okay, then shoots over here into the anterior pituitary. The cells of the anterior pituitary kind of interpret those releasing hormones that are released by the neurosecretory cells into the portal blood vessel, okay, and that triggers them to produce their own hormones, okay, and there are a few of them that you should know, okay, so one of them being TSH, okay, which you should know from the thyroid. FSH, sorry, that's hard to read there, and LH, follicle stimulating hormone and luteinizing hormone, okay, and these are going to act on either the testes or ovaries. We have GH or growth hormone, okay, so this is for growth and it acts on the entire body. It's no particular target cells. Okay, so that's growth hormone. And then the final one that you should probably know is called prolactin. That's an A, prolactin. Okay, and prolactin acts on the mammary glands, and we'll cover that in a later video. Okay, so just to review here, okay, hormones that are said to be produced by the posterior pituitary aren't really produced by the posterior pituitary. They're produced in the hypothalamus, in the cell body of these neurosecretory cells. They travel down those neurosecretory cells and are secreted into the capillaries of the posterior pituitary, and then they leave through that blood vessel. But they're actually, created up here in the hypothalamus. The anterior pituitary is much different. Releasing hormones, like gonadotropin releasing hormones, something like that, are produced in the neurosecretory cells of the hypothalamus, just like we talked about before. But those hormones then leave the hypothalamus through a portal blood vessel. That portal blood vessel then carries them to target cells in the anterior pituitary. Those cells in the anterior pituitary produce their own hormones, which then leave, and I guess I could draw this in, through their own blood vessel. Okay? So it's a good idea to also obviously know some examples of hormones that belong to each one of them. Okay, now let's return to our discussion on hemoglobin. And hemoglobin can often be noted, in this form. Okay, and HB4 basically means the 4 polypeptides that make up the hemoglobin. Remember, each polypeptide has one heme group. Okay, so hemoglobin with no oxygens is going to be HB4 and then nothing. This would be hemoglobin with 1 oxygen molecule, 2, 3, and 4 oxygen molecules. Okay, this is what we would call complete saturation. Okay, that the hemoglobin can't hold any more oxygen molecules. So let's take a look at how hemoglobin picks up and gives off those oxygen molecules in what we call a dissociation curve. On the x-axis of this curve, we have partial pressure of oxygen, and that is measured in mmHg, which is a common way that we see pressure, measured. And on the y-axis, we have percent hemoglobin saturation. Okay, and saturation refers to how many oxygen molecules that hemoglobin is carrying. Okay, again, this form over here would be completely saturated. Okay, the curve for hemoglobin looks something like this. Okay, so you're done basically with this graph once you're able to draw that curve. But just so we know, our typical range of partial pressure of oxygen is somewhere in here. So in typical body tissues, we never have such a complete lack of oxygen that we are causing hemoglobin to give up all of its oxygen molecules and become this form. That is very rare. Okay, and we typically don't have enough oxygen in our bloodstream, to push it all the way out here. There's no point in getting there because that hemoglobin is already saturated with oxygen. Okay, so most of the time we're going to be right in here. Okay, so the lower this pressure gets, the more likely hemoglobin is to give up one of its oxygens, which makes sense. The less oxygen that's in our bloodstream, the more likely it is that hemoglobin is going to give up one or more of its oxygens to that tissue. Now let's move on to something called the Bohr shift. And the Bohr shift basically measures the effect of carbon dioxide on hemoglobin and its ability to hold on to or donate oxygens. Okay, so again, I'm going to start off with the same type of curve that we saw in the last video, where we have partial pressures of oxygen on the x-axis, and we have percentage of saturation of hemoglobin on the y-axis. Okay, so again, this is going to tell us how easily hemoglobin is giving up those oxygen molecules. Now, if we think about why hemoglobin would be giving up or picking up oxygen molecules. There is a relationship there with carbon dioxide, and that has to do with, of course, cellular respiration. So you don't have to write this part, but just to give us an idea of where we're at here in our thinking. Okay, carbon dioxide is a product of glucose metabolism. So C6H12O6 plus oxygen yields water and carbon dioxide. Okay, and that's the overall equation for cellular respiration. So it makes sense to me that the more carbon dioxide tissues are producing, the more oxygen they're going to need. So it's kind of nice that the more carbon dioxide we have in a tissue, the more likely hemoglobin is to give up that oxygen that it's carrying to those respiring tissues. Okay, so let's just draw our normal oxygen dissociation curve. Okay. Okay, so this should look just like the last one. Okay, and this is going to represent when carbon dioxide levels are low. Okay, and carbon dioxide levels are going to be low when tissues are more at rest. Okay, they're not actively respiring so much. So we're not talking about times of exercise or any exertion. Now, when carbon dioxide levels are high, we experience what we call the Bohr shift. And that's going to look something like this. Okay, and so what this means is that I need less of a change in partial pressure to get hemoglobin to give up that oxygen. Okay, so ordinarily I would have to decrease this partial pressure all the way down here to get hemoglobin to give up that second oxygen probably. Okay, whereas if there's a lot of carbon dioxide in the atmosphere, I only need the partial pressure, okay, to be down here somewhere. Okay, so it's just requiring less of a change in partial pressure of oxygen in order for hemoglobin to give up some of its oxygen molecules. So carbon dioxide causes hemoglobin to donate or dissociate its oxygen molecules easier, and that's because carbon dioxide is produced by tissues that need oxygen because of their respiratory habits. Okay, now let's talk about the difference between fetal hemoglobin and adult hemoglobin. I'm going to start with, again, the same curve we've been looking at, where we have partial pressures of oxygen and percent saturation of hemoglobin. And again, that saturation refers to how many oxygen molecules that hemoglobin is carrying. Okay, so you don't need to write this, but this would of course be something like this: hemoglobin with no oxygen, and all the way up here at 100% saturation, that would be hemoglobin with 4 oxygens. Okay, anyways, we don't need that, so let's get rid of that. And I'm going to start off by drawing my normal curve, okay, for hemoglobin. And normal hemoglobin, of course, I'm referring to as adult hemoglobin. Okay, and so again, what this is saying is that I have to decrease the partial pressure of oxygen in order for hemoglobin to start giving up its oxygens to that tissue. Now, one of the times that we need hemoglobin to give up its oxygen is when a woman is pregnant. We need that hemoglobin that is coming through the placenta and giving the fetus its oxygen to be able to give up that oxygen to that fetal hemoglobin. And so that curve is going to look a little bit more like this. Okay, so another way of thinking about these curves is, of course, instead of giving up oxygens, okay, it also refers to something's affinity for oxygen. Okay, the harder it is to give up an oxygen, so in other words, the greater you have to decrease that partial pressure in order to give up an oxygen. We can also think about that in the opposite sense, okay, where the harder it is to get something to give it up, that means the greater affinity it has for oxygen. It's more attracted to oxygen. So when we see that curve shift to the left, okay, that means that this fetal hemoglobin has a much greater affinity for oxygen than the adult hemoglobin. So at the same partial pressure, okay, that means that this fetal hemoglobin is going to be able to steal, or rather it's this way, is going to be able to steal some of this adult hemoglobin's oxygen. And that's how we pass along oxygen from mother to child during pregnancy. That fetal hemoglobin has a greater affinity for oxygen than the adult hemoglobin. Okay, now let's take a look at our last oxygen dissociation curve, and this time we're going to compare hemoglobin to another oxygen-carrying protein called myoglobin. All right, if we look at these prefixes hemo and myo, hemo means blood. Hemoglobin is going to be found in our red blood cells. Okay, myo means muscle. This is an oxygen-carrying protein that is only found in our muscle cells. Hemoglobin has 1, 2, 3, 4 polypeptides. Each one of those polypeptides has a heme group, okay? And so that means I can carry a total of 4 oxygens, okay? Muscle carrying proteins, this myoglobin, only has 1 polypeptide, 1 heme group, and therefore it can only carry 1 oxygen. Now, in terms of its affinity for oxygen, or another way of thinking that is its ability to donate oxygens, it's quite different from hemoglobin. So let's take a look at this one. Again, partial pressure of oxygen on the x-axis, percent saturation. I'm not going to say of hemoglobin this time because we are, of course, comparing hemoglobin and myoglobin. All right, so let's go ahead and draw our normal curve for hemoglobin, looking something like this. Okay, again, this means that I have to decrease the partial pressure in order for some of those oxygens to fall off or get donated from the hemoglobin molecule. Okay. So let me go ahead and label this one as hemoglobin. Now, hemoglobin has 4 oxygen molecule carrying groups. So it's 4 heme groups. So I'm just going to number them. You don't have to number them. Okay, so like 1 would be here, 2 right at 50% saturation. Okay, that's not true. I don't know what I'm thinking here. Down here would be 0. Then 1, then 2, then 3, then 4 oxygens. Okay, myoglobin, on the other hand, can only carry 1 oxygen. So its curve is kind of an all or nothing curve. And it looks like this. Okay, so this curve up here is going to be myoglobin. Now, if we notice what's going on with this curve, as there becomes less and less oxygen in my tissues, okay, the first molecule to give up its oxygen is going to be hemoglobin. So at this partial pressure of oxygen, the hemoglobin starts to give up an oxygen. Here it's giving up another oxygen. Here it's giving up another oxygen. Myoglobin doesn't give up its oxygen until right before we're almost in an anoxic environment. So myoglobin is kind of the muscle's last resort. Right before it goes into anaerobic respiration, then this myoglobin will start to give up its oxygen molecule. But that's not going to happen unless we're in kind of an emergency situation. So this is really kind of a last resort molecule in our muscle cells that's holding onto all of its oxygen until we really need it, until it's almost an oxygen emergency.